I think we now continue with the poster presentations. In total, we have 6 poster presentations, but not everybody doing a poster presentation is already in our Zoom meeting. So if you are presenting a poster presentation and you are still in Vimeo, please go to the Zoom meeting location. Okay, I see already people coming in, so that's good. That's good. So what I will do is I will show the posters. I might use a slightly different order of the posters than was announced, because not everybody is here. So if you see your poster, please start talking and presenting it. You will have 3 minutes or so. Let's see. And Peter, we will start with your presentation. Hi, you can start.
Hi, I'm Peter Harnishman. I'm telling you a small. I'm telling you about the search of Bumblebee manicus subspecies dalmati last summer in the French Alps. I'm just a hobbyist, and I'm now pleased that I'm allowed to present this poster. I was triggered by a post on the Bumblebee Conservation Trust website about this bumblebee. It is a specialistic bumblebee foraging mainly on Cerynthe minor and Cerynthe glabra. It's present in the French Alps between 1,000 and 2,000 meters. And it's active early in the morning and late in the evening. To find this bee, I first plotted GPS locations on a paper map, and then we went to the French Alps, at first to Barcelonnette. We did multiple drives up Col d'Allo and Col de la Cayolle to find patches of Cerynthe, the main food plant. And as we were there, we found out that all plants had passed flourishing already. So nevertheless, I decided to take a chance and go up to Col d'Allo at dawn and at sunset, to be there for several hours to see if I could find the bee. But there were no Bumblebee manicus seen there. Later in the holiday, we went to the Queyras, even higher in the Alps. That's the region adjacent to Italy. The Queyras Risola Valley is at 1,650 meters. When we were there, we found a plot of Cerynthe just starting to flourish. And when we returned the same evening, we found one bird manicus bee sleeping in a plant, which was a great moment. The next evening, I went back, and the same bee, probably the same bee, was sleeping in the same plant at the same spot. And I've seen no other bees, so it's really rare. The next morning, it was windy and cold, 6 degrees centigrade. And I went back and found the same bee on the ground. And I decided to wait longer than I had planned, because it was told to me that the bee was active only 1 hour after sunrise. But because it was very cold, I stayed longer, and I found several bees foraging between 10 and 10:30. And I succeeded to make some photographs of it, which was very, very difficult, because the bee is changing between groups of plants with a high speed, and on a steep slope, it's really difficult to make photos. So now it was successful. It was nice to see, and this is what I wanted to share with you. I want to thank ICE for making this possible, and also I want to thank all the Bumblebee specialists sharing their knowledge so kindly to people who are just enthusiasts. And I think that is a very important thing in bee protection. Thank you.
Thank you. Let's see. Ruchi, you can start your presentation.
Okay, thank you, sir. Bumblebees are known as the important bus pollinators in the natural ecosystems. And as we know, that India is a home of 48 of the 260 known species. Bombus cimarrodalis is the native species of the bumblebee that we found in our local area. During the rearing of these Bombus cimarrodalis in the laboratory rearing, we found different pests and disease that resulted a negative impact on the growth and development of these colonies. Therefore, the present study was conducted to identify uh, these pests and diseases in laboratory and the field established colonies. During this research, a total of 130 queens were captured during the spring seasons and were kept in the BOD and the culture room under the control conditions for their growth and development. During their growth and development of these colonies, we identify four major pests. That is the conopid fly, Physocaplia tibialis, bee moth, Aphomia socialia, mites, that is the Parasiticus ficorum, beetle, Athena tumida, and a disease that is the Nosema bombai. And in our studies, we found that the maximum infestation was recorded for the Nosema bombai, that is the 54.7%. And these incidence was further followed by the conopid fly, that is 18.7%, and for other bee moth, and then mites and high beetles, that is only 0.7%. During the incidence of these pests and diseases, we also recorded the month-wise mortality of these Bombus hemerodalis queens during the growth and development of colonies, both in the laboratory and the field established conditions. And we found that the maximum infestation or in the dead queens, we recorded Nosema bombai infestation, that is 37.7% in the month of the August. As we also studied the effect of these environmental factors, relative humidity and temperature on these seasonal incidents of different pests and diseases. And we found a positive significant correlation of relative humidity and temperature for the Nosema bombai infestation. Therefore, the highest temperature and relative humidity conditions in the month of the August recorded for the Nosema bombai infestation. And for conopid fly, we recorded a negative significant correlation with the temperature. While for the wax moth, hive beetle, and the other mites, we found no correlation with these environmental factors. Therefore, to conclude my study, I would like to say that these environmental factors like temperature and relative humidity also showed a congenial effect on the growth of these pests and diseases, which later on hinders the growth and development of the Bombus hemerodalis queens for the colony growth and development. Therefore, we need to adopt some management practices for the to successful year-round rearing of these Bombus hemerodalis queens for the use of these colonies in the pollination that will be benefit the farmers and the apiculturists. Okay, thank you so much.
Okay, thank you. We continue with Florian. Oh, that's my mistake.
Okay, thank you very much. My name is Florian Schmidt. I'm a professor of evolutionary botany at the University of Zurich. First of all, I'd like to thank the organizers for this fascinating conference. It's really been a great series of talks. Now, my interest is in how pollinators and other interacting organisms impact the evolution of plants. And I've recently studied this phenomenon in a novel way, namely by using experimental evolution. And I'd like to present here the results of the first study done in this way. And here we have studied the onset of how plants adapt to different pollinators. And we have used bumblebees and hoverflies to do so. And experimental evolution works. We have set up populations of fast-cycling Brassicarapa plants. These are genotypes of Brassicarapa with a very short generation time, which is of course necessary for experimental evolution. And for 9 generations, the plants were pollinated either by bumblebees or by hoverflies, or as a control by hand. And then at the end of the experiment, the plants were phenotyped, so analyzed, and the phenotype was compared between these plants. And what we found is that the pollinators gave extremely strong selection and rapid evolutionary response by the plants. So the plants evolved strong changes with different pollinators, especially bumblebees, and that's the reason why I'm here. Bumblebees caused a lot of trait evolution in the plants. Plants became taller, as you can see in the picture here. This is the bumblebee pollinated plants and the hoverfly pollinated plants. So the plants were much taller. Keep in mind, those plants just had different pollinators. There was no difference in fertilization or light or nutrients whatsoever. Just the pollinators were different. Bumblebees also caused the evolution of more fragrant flowers. So flowers produced a lot more fragrance and also more UV reflection. So the color also changed. And at the end of the experiment, we also tested attractiveness, and bumblebee pollinated plants were a lot more attractive to bumblebees. So this was adaptive evolution and not drift. Because the plants increased their attractiveness to bumblebees. Hoverflies, on the other hand, were lousy pollinators. They did a bad job in pollination, transferred little pollen. And what they caused is not so much trait evolution, but an increase in selfing. So the plants pollinated by hoverflies had a reduced genetic self-incompatibility and produced a lot more seeds by spontaneous selfing. And the consequence of all this, I think, is that we have to be aware that changes in pollinator communities, for example, bumblebee declines, can have significant downstream effects on plant evolution. For example, we can imagine that with the reduction of bumblebees, hoverflies become more important pollinators, which are not as good in pollination, and then this may cause the evolution of increased selfing in some plant population. I think this is something to be aware of. This result is published in a freely accessible paper, so you can access this. It's published in Nature Communications, which is an open access study, so everybody can download the paper. Thanks very much.
Okay, great, thank you. Simad, please begin your presentation. Your microphone is still off, so.
I hope you can hear me.
Yes, we can.
Sure. Thank you so much, Vincent, for this opportunity. So my name is Schmidt Joseph. I'm with the Department of Entomology, University of Georgia. I want to recognize my colleagues, Karen Schulz, and also David Jesperson. So this is about turfgrass. So turfgrass is a very important component in southeastern United States. It's a very important component, and among different turfgrass genotypes, centipede grass is very common and widely found in this region. What the peculiar thing about centipede grass is that this grass flowers in the season, I would say late summer to fall. And we were curious whether how does pollinators interact with this inflorescence. So that's basically this work. We tried to do a survey type experiment here, and we went identified homeowners, basically residential turf grass, and we put out 3 sampling methods. One is basically sweep netting, where we went there and spent like 30 minutes, and if you see any insect on inflorescence for 3 seconds, we try to grab them with the netting. Other 2 methods we used was pan traps and melee traps. So what we found is basically in the sweep nets is that halictids, especially Lasier blossom species, was abundant, and also bumblebees, and that's why I'm here, right? So bumblebees were, I would say, 30 minutes time of sampling. It is quite significant, and we saw them foraging on the centipede grass inflorescence, and that you can see over here in this in the figure 1. There are other groups on honeybees, and also other aphids and halictids were also found. Within the trapping system, we saw more that the sweep netting is more effective. The pan trap and melee traps not very good for sampling bumblebees. So this is kind of a first report of showing that bumblebees are important players in centipede grass system, and that we want to explore further with looking at pollen loads and other fitness experiments. And that's all I got. Thank you.
Okay, thank you. So the next presentation is by Jose Montalva. Are you here already, Jose?
Yep, I'm here.
Okay, welcome. Hi.
Hello.
You can start your presentation.
Share it? Okay. Good morning. My name is Jose Montalva. I'm studying the bumblebees in Chile, South America. And this presentation is about the introduction and the invasion of Bombus terrestris, the European bumblebee in South America. Bombus terrestris was imported to Chile in 1997 for crop pollination. And as you see in the picture on the left, picture number 1, you can see the number of importation. In 1997, they start doing experimental studies on the Bombus terrestris. And if you see here in 2007, there is a spike of the commercialization of the Bombus terrestris for the crop pollination. Since 1997 to 2019, the number of importation reached over 1 million colonies and queens. You can see this picture over here was taken last year, 2020, and this is a high demand for Bombus terrestris for the crop pollination, especially for some plants like blueberries and other kind of these. And when you talk about the blueberries, there is not inside like tomatoes. Tomatoes at first they was used like in greenhouse for pollinate crop. But now the Bombus terrestris is using widely in a variety of plants like blueberries, cherries, and these open fields allow the Bombus terrestris escape to the environment. Since 2014, we started a project for tracking the naturalization and the invasion of Bombus terrestris, mainly in Chile. And we start tracking the Bombus terrestris by pictures, how I mentioned before in other presentations today, via citizen science. And you see here, this is our records from 2014 to 2020. This is the distribution of Bombus terrestris in the country from the almost all the country. There is you see here in the north part where it's mainly desert. You see in the frontier with Peru and Bolivia, and the picture on the middle figure 2, and so south like the subantarctic forest in Navarino Island. In 2006, the species invade Argentina, since they don't allow the importation of Bombus terrestris in that country, but they cross the mountains and now is present over there. When we talk about the invasion of Bombus terrestris, we know that it's affecting heavily this other bumblebee, the native bumblebee, Bombus dalvomi, the giant bumblebee. And we know there is, first we're thinking in the competition for resource, but now we know that there is many other, especially pathogens and diseases. Here is a picture of Cousinia laevis. Cousinia is a mite, it's not affecting so much the Bombus dalvomi, but this is the best picture I have. But there is another Nosema bombi, Critidia bombi, and Apicystis bombi, and this is affecting so much the population of Bombus dalvomi. There is now listed as an endangered species in the country. Thank you.
Okay, thank you. Andreas, you can start your presentation. Andreas, I can't hear you, so I'm not sure what's happening. Are you there or?
Hello, hello.
Yeah, that's good.
Oh, sorry, I chose the wrong microphone. I have two microphones. Hello, my name is Andreas Coulter, and I'm from the University of Gießen, and I'm a botanist, and my project is about time travel, basically. So we were comparing the nutrition of bumblebees from nowadays. So we went to the field and collected specimen, and then we went to natural history collections and also collected pollen from the bumblebees in the natural history collections. Of course, we didn't collect the bumblebees themselves. Sometimes we also collected one of the legs to confirm the species ID in the collections. And we did all of that together by meta-barcoding. So we isolated the DNA from the pollen, and then we sequenced it all together. And then basically what you can see on the right-hand side, sorry, it's a little bit small, but the posters will be circulated later. You can see like this pie chart, and it tells us which kind of species we found in the pollen basket. It's not quantitative, so we have to go by absence presence because of PCR bias and artifacts. And we went back in the natural history collections up to 40 years, but in theory, it is very well possible to go back further in time. The problem is that the DNA will get damaged and degraded. So you have to use a different molecular marker, and then you will be only able to get to plant species, to plant family level instead of plant genus or species level. And you need a very clean lab for that. And by clean, I mean like ancient DNA clean. There's a lot of potential in this method, I think, because it has a higher resolution than identification by microscopy. and it doesn't require as much manpower as observations in the field do require. But there are also some problems I would like to highlight here. One problem is that in the collections, there are very large gaps in space and in time. So when one of the collectors stopped collecting, there's like a gap of like up to 20 years until the next collector started collecting in the same area. So that's, of course, a problem. And the rare bumblebees, of course, it's very difficult to find them in the natural history collections. And it's also difficult to find them in the field, actually. We had a hard time, but we managed to catch a few rare ones. And our project was heavily focused on method development, well, fortunately or unfortunately, however you want to see it, because we found that most of the methods used for pollen metabarcoding were not really quality controlled very tightly, let's put it that way. So we were, for example, the first ones to use mock communities to verify our plant primers for universality, for example, and for taxa bias, which is also very important. Because, for example, we used the plant markers ITS1 and ITS2, and they are multi-copy markers that in theory should be all identical. But the number of copies per plant genome of these markers, for example, it varies between 100 and 100,000, which is really a gigantic margin. So you have to be careful not to drown out some of the species which have a low copy number in your PCR protocol. And in the metabarcoding protocol overall, it's also very important to pay tribute to the special characteristics of plants versus animals. For example, I can see a lot of protocols in the literature that use some kind of percentage threshold for identifications of genetic distances, which doesn't apply for plants because they don't really have like genetic gaps between the species in some of the markers we used. Which is also one of the things that can be improved in the future, just include either more markers in the study or just increase the read length. And thank you. Unfortunately, the paper is still under review, so I cannot present any results yet. But I think the results will be quite interesting. And thank you for your attention.
Okay, thank you. I think we reached the end of our two-day symposium. I would like for Linda to join me for this last few minutes. I want to thank all the people presenting posters or presentations last week or during this week. I think all the attendants liked the presentations, and the presentations were of high quality. What I saw in the chat is that most people were very interested and energized by everything we learned. So I think we had a very good symposium. So Linda organized this meeting, and I would like to have her on screen to say a thank you to her.
I'm here. Thank you.
Okay. Yeah. So yeah, it's difficult. We don't have a room filled with people, so we cannot have an applause or something. But I want to thank you for organizing this. I saw in the chat that people liked it very much, and I think we should contemplate of doing this maybe every 2 years or so, because there's lots of studies happening on bumblebees. Yeah, do you have anything to say as last few words?
Well, yes, I would like to thank the speakers as well, but also everyone who was here. And that's the reason why it was such a huge success, I think. There were so many applications. That was completely unexpected for me. I was just expecting maybe a few hundred people, but now we have more than 800 people who have registered. Not everyone was here on both days, but that is okay. I think a lot of people also watched the meetings afterwards. We will make them available on YouTube, and I will send around all the links and the links to the posters as well, so you can check them back afterwards if you want, or if you didn't have time to watch it the first time around. everything will be available later on. So thank you to everyone who contributed, and thank you to everyone who watched the presentations. And yeah, I hope you had a great time.
Okay, thank you, and have a good afternoon. And then we close this meeting. Goodbye.